Thank you, Paul, for introducing me, and it's now my pleasure to introduce Dr. Alexander White Zaranek, or Sasha, whom I know since 2006, as I met him as a co-founder in the very early days of the Harvard Personal Genome Project. We were walking together to the green tea line at Huntington, and then said, "Oh, I'm also in there from the beginning." Wow! So, and yeah, now it's 2016, and we're working together, and the Personal Genome Project has, say, grown beyond any expectations that we had back in 2006. We had one participant at that moment, so we couldn't know how it would develop. So currently, Sasha is a co-founder and the chief scientist at Curoverse, which is a venture-backed company focused on building free and open source platform for storing, analyzing, and sharing biomedical data. Sasha works on open technologies. That's also part of the tradition of the Personal Genome Project, of course, open data collection and open data sharing. Those technologies are part of the revolution that reduced human DNA sequencing cost by a millionfold since the completion of the Human Genome Project. So in the early 2000s, it was a million times more expensive to sequence the human genome than it is now. So this is unprecedented development. Sasha's current research focus is on the development of clinical quality applications for processing massive data. Data sets spanning millions of individuals across collaborating organizations. These are something like consortiums, huge collaborations, and eventually encompassing exabytes of data. And I must confess that I didn't know what exabytes were. I did just a test back in the lab and asked a very established colleague, and he hadn't heard of it, which made me very happy, but I'm sure Sasha is going to explain to you what exabytes are. And Sasha's contributions have led to highly cited publications in Science, Nature, and The Lancet, and other leading journals. And he's now going to tell you something about tiling the genome, naming the parts of the genome that make you, you. Sasha, floor is yours.
Thank you so much, Jeantine. Hi, everybody. So I'm going to start by showing you a piece of my genome. And here it is. You know, this is one little piece of it. If you look at the green dot, there is a C right here, where my genome may be different than most of yours, although there's probably a few people in this room that have that same feature in their genome. And this is very strongly associated with high cholesterol. And my mother had a tendency toward it. My grandmother did. It's a very well-established finding. We know that high cholesterol can be quite clinically significant. But interestingly enough, after much research and effort, it was found that people who do have this feature in their genome don't necessarily have a significant association with heart disease. So, you know, genomes are complicated. And this is really only about a millionth of my genome. We have an awful lot of genome to show you if I just sat there showing you slides of my genome. And you know, we happen to know something about this feature, but there's lots of features in genomes that we don't know so much about. Now, as Jeantine mentioned, we're both co-founders of the Harvard Personal Genome Project that George Church started in 2005. And so an interesting thing about my genome is that you can go to this URL, and it's completely public, and you can download my genome. In fact, there are several copies of my genome that were independently sequenced. And so if you need some data to play with, there it is. And I was actually just chatting with some members of the audience, and we have at least one high school student, not to signal you out. But obviously, if you're in high school and you want to study genomes, well, who's going to let you study really confidential data? so people like me who wanted high school students to be able to study genomes made their data public. And so I think it's very gratifying for me to see such a wide audience that isn't necessarily just biomedical researchers, because I think in the end, all of us have genomes, and all of us should be able to study them. And it would be nice if there was public data that you could play with that would give you kind of a sense of, like, what can you learn from a genome in a kind of hands-on way that you couldn't learn necessarily. And you may be very private about your genome. You might think that we public genome sharing people are crazy, but you would still benefit from having genomes to play with yourselves. Obviously, the world's biomedical data will mostly be private, but it's very helpful to have a collection of public genomes so that we can kind of get a feel for what's possible. So how are we gonna make sense of genomes? My genome, any particular genome. Well, the only way is to sequence lots and lots of people and to carefully phenotype those people, really look at their medical records, look at their history, their medical history, and amass all of that data together and to look at it in large cohorts. So we need lots of genomes to understand even one genome. And luckily, that's not too much of a problem, because over the next 5 years, we will sequence at least 5 million whole genomes and probably more than that portions or smaller portions of the genome. And this is a lot of data. It's about 500,000 terabytes. That's half an exabyte. And that's something, these days a little hard drive can easily store a terabyte. So 500,000 hard drives is still a lot of data if you want to share it with your colleague. So how are we going to do this? How are we going to amass these data sets? How are we going to make them useful? So the way that we're going to do it is we're going to build on the commons. And this is something that in IT, the Googles and Apples of the world have all been doing for years. So the most kind of well-known foundation in the space is called the Free Software Foundation. It's here in Boston. And their mascot is this little guy over here. The penguin over here is Linux. So if you have an Android phone, then it's powered by this guy. And if you have an Apple iOS phone, it's powered by the BSD daemon. They're both free software. At this point, the vast majority of computing devices in the world are all running free software. And this, as a business person, and a scientist, this is actually a big deal, because it used to be that we just couldn't imagine how you could possibly have software businesses that gave all their software away. But it turns out that over the last few decades, this has become very well established. There's billions of dollars of venture capital investment in IT companies whose products are all actually open source. Okay, so on the software side, part of building on the commons is this free software idea. On the data side, let's build on the Personal Genome Project. So at Harvard, these are the PGP-10. They're the first 10 participants in the Personal Genome Project. One of our goals has been to scale up the project when we could sequence people for about $1,000 per person. And recently, a company called Veritas that is co-founded by George Church and some of my colleagues has announced a commercial offering where you can get your genome for $1,000. They will give it back to you if you want, and if you want, you can donate it to the Personal Genome Project. You have to go through the Personal Genome Project consent process, and you really have to think about what you're doing and pass an entrance exam and some other things. But at this point, it is actually possible that we've hit this goal that we've had for a decade of the $1,000 genome. The other thing that's important for these kinds of scientific studies that the Personal Genome Project offers is that we offer cells. So if you're a biomedical researcher and you need an actual tissue sample, then participants that want to donate those tissue samples can do that through the PGP, and there's various biobanks that will store those tissue samples and make them available to researchers. And the most successful example of this at this point is the National Institute for Standards and Technologies has chosen 2 families from the Personal Genome Project, a mother, father, child, 2 trios, to be kind of the reference material genomes. So those are now very widely available, and if you are a clinical lab and you want to sequence a genome, you can order these materials from NIST and use that to validate your sequencing so you actually know that your sequencing process is good and not just nonsense. So PGP has already really made quite a lot of impact with an incredibly sort of ardent but small volunteer staff. So the questions I've been thinking about with my colleagues at PGP, with my colleagues at Curoverse, with students, have been around this question of, well, how do we organize the sort of computational resources so that when a scientist asks a question, when a member of the general public asks a question, or when a physician asks a question about a genome or about genomes in general, that we can actually answer those questions. Because obviously, as I sort of alluded, that there's going to be 5 million genomes, but those 5 million genomes are going to be spread all over the world. It's a huge amount of data. It's all locked in little silos. How are we going to ask questions of these data? And if I ask a question of your genome, how do I know that when I'm asking you about a bit of your genome, that we're talking about the same part of your genome? This is where the title of my talk comes from, is that we need all of this infrastructure to make it possible for us to share, but we also need to just be able to make sure that we're talking the same language. So I'll give you an idea of how we do that. So what do we have now? Well, this is these hard drives, sit on benches pretty much everywhere that that sequences genomes. Genomes are often delivered as hard drives. And this is quite an old picture. And so, the hard drives have gotten a little bit smaller and they store a little more data, but it's still the same horrible mess that it's always been. That, if I gave you 5 million of these and told you, well, I need to ask some simple question, it takes an hour just to load one of these hard drives onto your computer. It's really totally impractical. And also they fail and they overheat and you lose data. It's really terrible. So we need some kind of platform. We need some big standard platform that people use that make this data available, that can be everywhere around the world, sort of a planet scale. And the features I think about that a platform like this has to have are these 4 things. And these are really unique because some of you may have, you may know that big data is something in the news all the time now. There's lots of companies that are creating products for big data. But biomedical big data is actually, despite all of its similarities with the rest of the big data world, does have interesting, unique features. And so these are 4 of them. So one of them is that in biomed, we need to share more than in other organizations. Our regulation and our security requirements are among the most complex in the world, especially in tension with our need to share. Because obviously sharing and privacy are in some sense in conflict. We also need provenance and reproducibility. And the data is unique. I showed you a piece of my genome at the very beginning. We're going to talk about that a little more. But the vast majority of my genome is exactly the same as the vast majority of your genome. So it doesn't really make sense to not have very special, if we're going to store exabytes of this data, then we might as well have very specialized systems that are super good at handling the details of this kind of data. So sharing. How should sharing work? Well, today, when Google and Facebook run their companies, they build data centers all over the world that have very large scale. And those data centers cover the globe in a kind of planet scale computer. And when something needs to be computed, you move the question to the data. And we should be doing that too. So if I'm here at Harvard and we have several data centers at Harvard, there's one at the Macy's building actually downtown. For most people, we're unlikely to know that. But there's a very large data center downtown. And so you want to be able to move the question to that data center. And if you have a collaborator in the UK, then you want to be able to move the question to that data center. And you don't want to say, hey, please ship me some hard drives with genomes on them. That's what you don't want to do. And of course, federation can be optional. Your data is very private. You don't want your doctor to share it with anybody, but you do want your doctor to have it. So there's no need for federation if that's your preference. Now, Mark Zuckerberg and Larry Page, they have these planet-scale computing systems, but they don't have a need to share the way that biomed does. They don't share their customer records with each other. They don't really share much of anything with each other, and they don't have to. But in biomed, we do need to do that. Now, I could spend this entire talk talking about governance of data. I think the only thing I'd like you to appreciate, because I'm a person who's comfortable making my data public. But before I became a biophysicist, I was a cryptographer. And I am very conscious about privacy and security. And I want to know, if I promise you that I'm going to keep your data private, that I really mean it. And so I would like those promises to be based on some kind of public scrutiny. So the systems that keep your data private, because your preferences are to keep your data private, should be based on real-world systems that run on public data, where people like me and others have made their data public, and so you can be assured that people, cryptographers, security experts, have actually tried to sort of crack into those systems and really tested how secure they are. So even though I'm talking primarily today about public data and open source software, the reason I care about these things is so that your data is private, if that's what you want. And so it's something that is really a very important principle, that even though you yourself might not be a cryptographer, you can go to people that you trust who are authorities on analyzing cryptographic systems, And you can say, hey, should I trust these people with my data? And if all of the systems that have your data are open source, then they can be audited. And you can get that kind of answer. If everything is proprietary, then you sort of have to trust the company that they're doing everything that they say that they're doing. Okay, so Provenance, this is Charles Darwin. Obviously, all life on Earth is connected through an evolutionary process. And when we're doing these very large computations, we want, especially, it's hard to appreciate really how big an exabyte is, but it really is quite big. So if you are doing computations at that scale, you want to know that those computations are reproducible. Could you do that computation twice and get the same answer? One of the things that computer scientists are used to when they work at this scale is that unlike your computer at home, which probably will work, and if it doesn't work, you'll just turn it on and off and try again, computations at the exascale are guaranteed to have computers that break. So you can't just turn off your million computers and turn them back on. You have to be tolerant of failure. And so provenance gets very, very important. You really want to be able to know the history of a computation, how it was done, and so on. So hopefully this is an amusing slide. So this Paris Hilton up there, she's the blue on this graph here. So she was much more important. The red line here is searches for cancer, and the yellow line is searches for medicine. So Paris Hilton was much more important than cancer and medicine, at least several times, in the history of the internet searches. But on the whole, cancer and medicine are pretty important. And so what I'm hoping to get you to think about is that as an internet company, Facebook, Google, really the biggest commercial enterprises responsible for building this kind of planet-scale infrastructure that I'm talking about, their use case is people searching for Paris Hilton. And so in biomed, we care a lot more about explaining, why did your doctor make this decision based on this report they generated 10 years ago? Recreating the exact search for Paris Hilton from 10 years ago, far less important. And so provenance is just much more important in biomed than it is in the internet world. And I've already mentioned unique data, but it's just a fact that all life on Earth is ribosome-based. There are very small differences in the ribosome across organisms on Earth. And so everyone has a genome. All life on Earth has genomes. And ultimately, they have proteins. And so this whole sort of mechanism of life on Earth has a huge amount in common. We have a lot in common with each other, and we have a lot in common with other species. And so you don't have to store every little bit of that exabyte. You could potentially compress the data enormously if you take advantage of the fact that this data is so unique. So when we weave all of these together, at the Personal Genome Project, we built something called Arvados, which is a platform, if you want to call it that, that's open source, that includes all of these components. It's been running on systems like this. It runs in the cloud. So it runs on Amazon Web Services. It runs on Google Cloud Platform. And it's the thing that powers the Personal Genome Project, and it can power lots of other things. But the idea is that because Arvados is open source, we can run it everywhere. We can run it in the cloud. We can run it in private data centers. We can use it to stitch together this sort of planet-scale computer that I've been talking about across organizational boundaries. This diagram is not important, but But the arvados.org at the bottom, the URL, is important. There is a very vibrant open source community around this project. The Sanger Institute in the UK, all sorts of pharmaceutical companies. Lots of people are really starting to realize, why are we all recreating this infrastructure? It's completely not competitive. The pharma has called us pre-competitive. But it means that, I had a high school student actually for 2 weeks who just finished last week, and so they were using Arvados and they were contributing to Arvados, and I didn't really need to make them sign any super complicated agreements because it's all open and public, so you can just go and contribute to it. If you have a niece or a nephew that wants to play with this stuff, this project is pretty unique in terms of making that possible. Without normally, the sort of onerous legal agreements you would expect working with, Harvard Med or something like that. So a component of Arvados is this part of the software that handles genomes, that names parts of your genome. The system is called Lightning. The idea is that it's very fast. It operates in real time. It will actually be able to answer questions in less than a second. Over time, we hope it will do that at planet scale. So your doctor could ask a question on their mobile device, and genomes would be interrogated all over the world. And not just genomes, but other kinds of biomedical data. And you don't want to just ask, do other people have exactly this feature that I'm interested in? But you also might ask for things like, are there other patients that are similar to my patient? And those kinds of queries in the past, the way it would work is actually the way it works in clinical genetics is that all the clinical geneticists know each other. There's only maybe a few thousand. And they get together at the same conferences. And so when they see a patient that looks like another doctor they know might have a similar patient, they just call them up on the phone or they email them and they say, hey, do you have a patient so-and-so with these features? But this isn't going to work, right? I mean, you can't rely on phone calls and emails now that we're generating millions of genetic features for every single person. It just cannot possibly work. And even in the old world, the doctors could talk to each other and they would eventually realize they're talking about the same genetic feature because Dr. A would call it one thing, Dr. B would call it something else. And so, in the course of the conversation, they would come to an agreement, oh yeah, we're really talking about the same thing, or oh, actually we're not talking about the same thing. Again, can't do that if we really are working at this scale with millions of features per patient. In the process of building Lightning, it's worth keeping in mind the doctors are doing clinical genetics today, and they've been doing clinical genetics for decades. And there are standards organizations that have been around, I think, since the 50s, actually, in the field called cytogenetics. And the way what these pictures are reminiscent of is actual microscope staining of your DNA. So you take your DNA, you put a stain on it, you look at it under the microscope, you try to stretch it out so it's kind of straight, and it looks kind of like these pictures. So you get bands that are dark and light depending on where the stain changed the color of your DNA. And so that nomenclature has been around for decades, and so part of the design goal of Lightning is we need to make sure that when a doctor asks for a bit of genome using the nomenclature they've been using for 50 years, that Lightning knows about that nomenclature and that it can talk to doctors in their language. So this is a pretty busy diagram. But I'll try to explain a little bit about how Lightning works and how we name bits of your genome. So the important thing is this unique 24-base tag sequence. So the idea is that the way we name genomes, the way we decompose them into little bits that we can all talk about in a consistent way, is through the standards process, we pick about 10 million 24-character sequences that are unique, and we use them as landmarks. And once we've picked those sequences, we call them tags. Then we can decompose your genome into what we call tiles. And every tile in your genome can then be given a unique name. And I'll show you some examples, but just show you in this diagram. So this is a tag, it's 24 bases, and then this is some sequence in between that's 200 characters, just whatever happens to be in your genome. Then there's another unique tag, and so forth. So your whole genome gets tiled in this way with the little green boxes are the tags that delimit your genome. And you have a copy from mom, let's say on the top, and then you have a copy from dad on the bottom. So you have two sets of bases that are very, very similar. So in total, you have about 6 billion of these characters in the ACGT alphabet. And so that's what your genome is. And over here, this little yellow square, this would be a position where this person has a sequence that isn't the common sequence. It's different from kind of what most other people have, especially most other people of their ethnicity, because there's a lot of variation is very specific to different ethnicities. And then the next encoding is just another person, and you can encode millions of people this way. And the nice thing about this encoding is that it's super compact. So the raw data for a genome is 100 gigabytes. It takes one of these big, huge hard drives. I can encode sort of pointers into this library of sequences of tiles in 4 megabytes. So 4 megabytes you can attach to an email. You can send it around. And for me, this is slightly amusing because there was a scientific paper about genomes as email attachment. And George Church, pretty much as soon as the paper came out, was like, yeah, genomes are 4 megs. It's really no problem. Everything is solved. We don't have to worry about these huge 100 gigabyte data sets anymore. And I would argue with George. I would say, no, George, that paper doesn't do this, and it doesn't do this, and it doesn't do this other thing. We can't use that. I'm not even sure it's physically possible to make a 4 megabyte genome. But last year we published a paper with sort of 20 megabyte genomes, and now we have 4 megabyte genomes. So George was right, and I was wrong. But so it is, in fact, possible. So let me show you how this works with my genome. So here's, you can think of this as a tile, right? This is just like a big tile, right? Lots of A's, C's, G's, and T's. So now I've added tags. They're blue and underlined. So there's not actually one tile here. There are 7. And then I'm just going to reorganize it a little bit. All the A's, C's, G's, and T's are exactly the same. I've just moved stuff around so that the tags are in a column. And so each one of these things is a tile. And the important tile, the one that was associated with cholesterol in my genome, is this one right here in the middle. And it's a little red oval. So this is the same sequence, the one in the middle. And this is not, there's no fun punchline here. This number is 32 characters. It's a unique, consistent name for this tile up here. And the nice thing about this number is that if you have that number, it means you really have this feature that's associated with a cholesterol. It's completely unambiguous. If 2 different labs around the world sequenced your genome and my genome and somebody else's genome, and they came up with this number, then you would know that we had the same feature in our genome. And that's not the case today. Today, you get a pile of ACs and Gs and Ts on your hard drive, and you might get something called a variant call file. And those files can be very ambiguous. And so it's actually very, very easy to end up in a situation where you're trying to figure out, well, does your patient really have this bit of genome? No, maybe not, maybe. And so this is I'm sorry, this is a very technical procedure, but it is something that allows us to actually do this kind of sharing. And over time, we'll give these things nicknames and human-friendly names, but for now, it's good that the machines have a consistent way to name bits of your genome, because otherwise it wouldn't be possible to automate these things that our doctors were doing just by phone calls and emails. Diagram is from our paper. At the top is kind of a traditional view of a chromosome. In the middle, there's a kind of a graph structure. Genomes, you can start thinking of them as making graphs, where the tiles in a person's genome represent all of the one path through the graph, and then all of the different people in the population have different paths through the graph. And then this particular thing here is just like the tile I showed you from my genome. This is from a PGP participant's genome. This is very strongly associated with breast cancer. We haven't actually established that she is aware that she has this. She had breast cancer at 66. So, from her perspective, she already knows she has breast cancer. But we did notify participants about this, and sometimes participants get back to us, sometimes they don't. So that's part of the nature of this kind of public research. But this is an actually clinically very important variant in a genome. And again, you see this string 355-2B9. This is a computer-generated string that is unique and completely unambiguous in terms of identifying the feature in this person's genome. The nice thing about this encoding, so this is called principal component analysis. This is a very simple thing that people do when they talk about machine learning. So if I give you lots of genomes and you apply this sort of statistical technique on those genomes, then what you'll end up with is people will separate by ethnicity. So all the people who are Caucasian will end up in one pile down here, and people who are Asian and are over here, and people who are African are over here. And it's kind of what you expect, that we have a huge number of features in our genome that are very similar to people of similar ethnicity, and so you get this kind of separation. And this tells you that our sequencing is working and that it's not full of sequencing errors. But this is a very rudimentary form of machine learning that the encoding I mentioned is not only compact, but it's extremely convenient for applying machine learning techniques to genomes. And I don't know if anyone saw the news around this. I mean, probably people know that IBM Watson defeated the Jeopardy champion. And people know that IBM also beat the world's chess champion. So recently, an actually much harder victory was achieved by Google. So AlphaGo defeated the world's best Go player, which I think is probably the hardest board game that we have in the world. So machines are just getting really good at doing things that human experts have been good at. And so I think we can expect that machine learning is going to have all sorts of breakthroughs in biomedicine over the course of the next few years. And these kinds of encodings and consistent naming schemes are going to be essential to make that happen. So again, Lightning is federated. So the idea is that you can send these queries all over the world. Your doctor is working on their little smartphone, and queries go out to data centers all over the world, and an answer could come back immediately. And initially, this is not going to be deployed in every physician's office. It could be a decade before this happens in sort of routine care. But for pharmaceutical research, for drug discovery, and also just for general discovery by the general public, I think that people will want to play with this data as it becomes more available. People who have played with looked at ancestry or other, predicting eye color, blood type. There's just lots of things that just pop out of this data. And so I expect that people will want to play with their own data. Hopefully, I know this is it's a very dry subject. This is the infrastructure behind genomics. And so all of it, though, although it may be very dry, it is accessible. And so my hope is that even if it isn't something that you think you will do personally, that you do reach out to your nephews and nieces and young friends and invite them to participate in this very open community. Because you don't need a PhD, you don't need special secret top security clearance because the code is public and the data is public. And this is one of our meetings at the Personal Genome Project. I think Jeantine is nicely featured, and I'm there as well. So thank you guys all very much. I hope I can answer some questions.